All right, so let's go ahead and begin. All right, assalamu alaikum, everybody. This is Biology 2.03. All right, so first of all, take a look at this screen over here, right? All right, Anas, are all of these things made out of cells, or some of these things are not made out of cells?
Some of these things are not made out of cells.
All right, so name something here that's not made out of a cell.
Oh yeah.
Atoms.
Atoms. Yusuf, what else?
Viruses, brother.
Viruses, right? So viruses are not cells. What about trees?
Trees are made of cells, brother.
Trees are made of plant cells, right? Right. So some of these things are kind of like usually like parts of a cell, or viruses are a little bit different from cells, right? Um, so some of these things are made out of cells, and some of these things are not made out of cells, right?
Mhm.
Right. All right. So today we're going to talk about cells and like cell theory and like the history of cell theory, stuff like that, right? So what is cell theory? It's just the idea that like things are made out of cells and they have certain characteristics, right? So we have like a couple of different things here, right? So it says all living things are made out of cells. So for example, a virus, right, is not made out of cell. So is a virus considered to be alive or not alive?
A virus is considered to be not alive, brother.
So, so not alive, right? So that's why a virus is considered to be different. It's considered to be not a living thing. So bacteria have cells, right? So there's different kinds of cells, right? So some cells are more complex than others. However, uh, but then like viruses are not cells, so therefore they're not considered to be alive, right? So this is the
Mhm.
basic unit of structure and function in all living things, right? So you can divide things until you get to a cell, and then after that, it's difficult to kind of divide it and keep that thing sort of alive, right? Also, new cells can only reproduce from existing cells. So let's suppose you have. So we're gonna look at some experiments that kind of show that, right? So that if you want to create like a new cell, you have to take a previous cell and then cause it to replicate in order to create, um, you know, another cell. All right. So these things right here, over here in this picture, these are just some additional examples of things from cell theory. But these are the basic, ideas of cell theory, right? All things are made out of cells. It's the basic unit, and if you want to make a new cell, you have to go back and reproduce a previous cell. Turn the light on. Yeah. All right. So this just we're going to talk about some of the different scientists, right? So we already talked about this guy Van Leeuwenhoek or something like that. So he created like the first. He produced like the first type of microscope, right? So we talked about this last time, right? So he observed like microscopic organisms and cells in all different kinds of things. He looked at like water and found all kinds of tiny like animals and or protists and things like that in the water. So in this one, right, he used sunlight, right? So on this sample holder on this pin, he would put like a drop of water or something there, and then he would look at it through the lens with the light coming through from the sun. All right. So we talked about this a little bit last time, right? So then we have Robert Hooke, and he came up with this idea of cell theory, right? So you see these tiny cells over here. So these are from cork cells, right? So these are from cork cells, so from the cork plant, and this is like the bark. So he looked at it and he observed all of these tiny cells, and so this is how he came up with the idea that okay, cells are like the living individual units of living things. Right. Now this person Robert Brown came up with the idea of the nucleus. Right. So before we knew there were cells, right, but now we got to figure out okay, what are the different parts of the cell? So he observed these central structures in plant cells and he called them the nucleus, right? Like in an atom, right? We also have the nucleus, right? So nuclear just kind of means like the center of something, right? So this was this person Robert Brown. So he came up with the idea of the nucleus. All right. So we have this person Schleiden, all right? So he published a conclusion stating that all plant tissues are composed of cells, right? So he wasn't just saying that there are cells. he's now we're starting to get the idea. Okay, like pretty much everything that we're looking at, right, has cells, right? So all plants that he observed have cells, right? So all plant tissues are composed of cells, and that an embryonic plant arose from a single cell. So
Brother.
Mhm.
All living things are composed of cells, right?
Right. But no, but so at this point, this person Schleiden is only talking about plant tissues, right? So at this point, we don't have like a good cell theory yet, right? Right now, they're still just kind of looking at a lot of things and saying, oh, we're seeing a lot of cells all over the place. So this person Schleiden says that all plant tissues are made of cells. He's not talking about animals and other things. He's just talking about plant tissues, right? So he basically decides, okay, plants are made of cells, and that you know a plant that grew, right, came from an original one cell, all right. So he declared that the cell is the basic building block of all plant matter, right? Only of plant matter. So this was the first kind of sort of generalization, right? The first time that someone came up with the idea that okay, there are, cells are like, it's not just that we observed some cells somewhere, but that everywhere we look, this is what we're seeing. Right? Now, this person Schwann came around and said that okay, all animals are made up of cells, right? So Schleiden said this about plants. This person Schwann said this about animals, right? So that's why they often say that Schleiden and Schwann, they came up with cell theory, because one person said about plants, one person extended it to animals, all right? So he basically came up with these two statements, right? Cells are organisms, and all organisms consist of one or more cells. So basically, organisms, like what are they made of? They're all made of cells. And the cell is the basic unit of structure for all organisms. So this is the first kind of official cell theory kind of thing that came about, right? So this is the basic cell theory that Schwann came up with, right? So Schwann, you may have also have heard of Schwann cells. So this is named after him. So this is like a neuron, right? This is like a brain cell, not in the brain. This is like in like a nerve in your arm and stuff like that, right? So these cells that surround the neuron, these are called Schwann cells, named after this person Schwann, right? All right. So this person discovered that reproductive cells are also cells, and then Virchow came up with this idea that spontaneous generation does not exist. So what is this idea of spontaneous generation? It's the idea that like cells can come out of like just from anywhere, right? Or the idea that living things can just come out of the ground some random place. So for example, like, so let's suppose you left like some meat somewhere, right? You left like a piece of meat or something rotting somewhere, right? Like a dead animal. Well, pretty soon you come back again, and you would see that there's like maggots and flies and all kinds of stuff there, right? There's like a bunch of maggots crawling out in and stuff like that. So some people, they thought, oh well, these living organisms, the worms and stuff like that, they just came out of the meat somewhere, right? Like they just sort of appeared out of the meat. So he did this experiment where he would put meat into different jars, and what he found was that the maggots only appeared when the jar was open. So what does that mean? It means that like a fly came and laid eggs, and then that's where the maggots came from. But if you close it off,
Oh yeah.
then what happens, right? The flies can't get in, can't lay the eggs, so you don't have just like spontaneous like worms and stuff coming out of the meat or coming out of something rotting, just lying around, right? It has to come from another living thing. Does that make sense, right? So this idea that living things can come out of just some random dead thing is called spontaneous generation. So this person Virchow, right, is saying that cannot really happen, right? So this is the experiment that he's using to kind of show that.
Yeah, I understand. What did virtue think?
So wait, in this experiment, right? He's showing that, okay. So which of these flasks shows like maggots, like worms, on the meat?
Unsealed flask.
Unsealed flask, right? In the middle one, right, you don't see any worms or maggots. So what does that show? It shows that the maggots, like, they're not just coming from the meat itself. Like a fly is coming and laying eggs, and then from the eggs, the maggots and worms are appearing. So before, people thought that that maggots just came out of the meat itself. Like if you just left some meat and it started to rot, that like maggots would come out of the meat, not like flies were coming in and laying eggs on the meat. Right. So if you seal it off, right, if you seal off the container, then you don't see any maggots. This shows that the maggots, they're coming from the outside somewhere. They're not coming from just the meat itself, just from something rotting itself. All right. So this is another test of spontaneous generation. So there's another scientist called Louis Pasteur. So he came up with the idea of like pasteurization, right? When you see like, oh, this milk has been pasteurized, right? That's to kill off the bacteria, right? So you just heat milk up to, or anything else like orange juice, to really high level for a very short period of time, just like very quickly, and then bring it back down. So that kills off all the bacteria, and so therefore people don't get sick from like bacteria from milk and orange juice and stuff like that. So, but so apart from the pasteurization stuff for milk and stuff like that, he conducted a test of spontaneous generation, right? So he had two kinds of flasks, all right? One of them, right, is basically kind of like open to the outside, right? So both in both of these are open to the outside. Air can flow in, right? In both of these, air can flow in. However, this one on the right, right, is basically just like a straight shot right to the air, right? In the other one, the air has to sort of like curve down below and then come back up and into the flask, right? So air is still circulating through in this S-shaped flask, but the air has to kind of like curve down below and then come back up like that. So what is the difference, right? What he found was that in the ones that are open, these ones to the right, bacteria started to grow. In the ones to the left, bacteria did not grow. So does anyone know like why that happened? Why is it that bacteria started to grow in these ones over here on the right, but in this one on the left, the bacteria did not grow, even though air can come into both of them, and bacteria is coming from the air.
Brother, I would assume that microorganisms are also subjected to gravity, brother. So like they somehow just like fell in, you know what I'm saying?
Mhm.
Like they fell in.
Okay.
While the curved one, they can't really fall in because like it's curved, so they
The
can't.
curved one was
go through.
boiled too,
you go.
so there was no extra bacteria.
Both of them were boiled. Both of them were boiled.
Yeah, both of them are boiled, right? All right. So Freya, what do you think?
I was saying because of the opening, like
So because of the opening, like what's going to be different because of the opening? Like how does the different openings change?
for the one without the magnet, like it's directly like
Mhm.
it could like go, but for the curved one, it has to go like through a more complicated way.
Right. So back to what happens on the one on the right is bacteria can just fall in, like Yusuf was saying, right? It just falls in. In this one, in the curved one, when bacteria kind of like falls in, it settles in this curve at the bottom. You see this curve at the bottom? The bacteria falls in, right? But bacteria is not the same as air, so it's not gonna.
I was right, brother.
It's not gonna.
I was right then, right?
Yes, you're right. You're correct.
Let's go.
All right. So yeah, scientist Yusuf over here, right? So the bacteria is not going to go down and then come back up and defy gravity, right? So bacteria is going to fall down, but it's going to collect in the bottom of that flask. So it's not going to be able to continue down and into the broth and grow. So this is also a test for spontaneous generation, right? It's kind of showing that look, even though it's connected to the air, both of them are connected to the air. Bacteria, right, only grew in one of them, right? So bacteria is not coming out of the broth itself, right? It's not coming out of the growth medium itself. It's coming from the outside, right? So this also shows that there's not spontaneous generation, that if you want a living thing like bacteria, you have to get it from another bacteria from before. All right. So there are two main kinds of cells. One is called eukaryotic cells, and the other is called prokaryotic cells, right? So prokaryotic is basically kind of like the more simpler kind of cells, right? Prokaryotic is more simpler, and eukaryotic is very complex. So like animal cells, plant cells, all the cells that make up humans, tigers, oak trees, pine trees, all those kinds of things are all eukaryotic cells, right? So prokaryotic cells are bacteria. There's another group called like archaea. It's basically kind of similar. So these groups, they're very, very simple, right? The cells are usually much smaller. There's not as much complexity in them, all right? So we're going to talk about what the difference is. So one difference they're mentioning here is that a prokaryote doesn't really have a nucleus, all right? So the DNA in a eukaryotic cell, right, in a plant cell, animal cell, the DNA is kind of sequestered in the middle and kind of protected from the rest of the cell, right? That's in a eukaryotic cell. The DNA is, is by itself. In a prokaryotic cell, the DNA is not protected. It's kind of floating around in the cell, all right? So in the prokaryotic cell, the DNA is not separated from the rest of the contents of the cell. Right?
So, brother, the difference, one of the differences between prokaryotic and eukaryotic cells is that prokaryotic cells do not have a true nucleus,
Correct.
and eukaryotic do have a nucleus, right?
So eukaryotes have a true nucleus. Prokaryotes do not have a true nucleus. Right. So this is just like another. This is just like a picture, kind of showing, that a eukaryotic cell on the right is much more complex. On the left, it's much more simpler. You can see that on the right, right? In a eukaryotic cell, the different parts of the cell are kind of organized better, right? Things are separated out. It's like this, like, for example, so you can think about it this way, right? So, the prokaryotic cell is like the TU building, right? And then the mushed is like the eukaryotic cell, right? So in the mushed, right, like all the classrooms are separated by walls and stuff, right? It's like a true separation, right? Like each classroom is like separated. There's like a wall, there's a door, everything to go in and out. You have to have a specific, you have to go through the door. It's closed off, right? Each section, right? The different parts of the cell, the mitochondria, the ribosome, this and that, they're all separated out by membranes. In the TU building, right? It's kind of like the whole TU building is like kind of divided artificially into like kind of like multiple classrooms, right? But the whole thing is really one big place, right? So in the prokaryotic cell, you don't have those divisions within the cell. You don't have like specific places where, like, okay, my mitochondria is here where I'm gonna make energy. Then the place where I make protein is over here. It's all kind of mixed in together. Does that kind of make sense, right? So in the eukaryotic cell, that's called membrane-bound organelles. Each organelle, each part of the cell, is kind of bound. It's kind of surrounded by a membrane that separates it from the rest of the cell. In the prokaryote, you don't have those. You don't have those membrane-bound organelles. Right? So these are just like some examples between eukaryotes and prokaryotes, right? So prokaryotes are very, very small. Eukaryotes are much bigger, right? That's why prokaryotes, like bacteria, when you see them in pictures, they're like very, very small compared to like a big animal cell, right? So eukaryotes have a membrane-bound nucleus, right? But prokaryotes do not have that. Now in eukaryotes, they also have, their DNA is much more complex as well, right? So the DNA is also bound up with all kinds of proteins and different things in there that helps to kind of compact the DNA. In a prokaryotic cell, the DNA is just like a circular kind of thing. All right. So there's other kinds of things like that, but those are kind of the main differences between eukaryotes and prokaryotes. So both of them in the center, right? You can see both of them have flagella. Anyone know what is flagella? Anyone heard this term flagella before? All right, Freeha, do you know what is a flagella? You're not sure. So flagella is basically like a long whip-like thing that cells use to swim. So bacteria cells, they often have flagella, right? Both of them around the outside have a plasma membrane, right? So both of them have a plasma membrane, right? Now, in a prokaryote, pretty much all of them have cell walls, right? In a eukaryotic cell, only the plant cells have cell walls, or some other types do also, but usually plant cells have cell walls, and animal cells do not have cell walls, right? So in terms of both of them have cell membranes, both of them can have cell walls, but even though both of them can have it, generally in a eukaryotic cell, it's going to be like more complex. So it is present in a prokaryotic cell, but it's usually not as complex as in a eukaryotic cell, right? All right, so there are other similarities as well. So both of them have ribosomes. All right, so there are bacterial ribosomes and eukaryotic ribosomes. Anyone know what do ribosomes do? All right, Anis, have you heard the term ribosome before? Or not as much. So ribosomes, they basically make proteins, right? So ribosomes, they're going to make proteins. So both bacteria and prokaryotes and eukaryotes, they're going to have ribosomes, right? Right. So we talked about this before, right? So in prokaryotic cells, you're not gonna have specific, membrane-bound organelles. Like everything in the cell, all the enzymes and stuff, they kind of like mix together in a prokaryotic cell. You don't have specific divisions where certain kinds of proteins are put over here, other things are put over there, a third thing is put over here. Everything is kind of mixed together, right? But in a eukaryotic cell, things are divided out. Those divisions are called organelles, right? So if you've heard of things like the ribosomes,
So whether the things in prokaryotic cells are not called organelles, even though they're not membrane bound.
right? So in prokaryotic cells, they do not have organelles. Yeah, yes. So organelles are membrane
They
bound.
have or
They do not have.
so they do have like things that help them like function and stuff, but they're just not called organelles, right?
Correct. Yep.
All right.
So prokaryotics still respond to the environment and reproduce. So we call them simple, right? But compared to anything that a human being can make, right? Even like the simplest bacteria is like incredibly complex, right? So it's almost like this tiny microscopic robot that goes around and can sense the environment, figure out where food is, can swim towards, if it figure out, oh, there's more food chemicals coming from this way, it can swim towards it, all kinds of things like that. There are also prokaryotic cells, like this thing called cyanobacteria. So on the right over here, this is called cyanobacteria. So these actually, perform photosynthesis, all right? So they also produce their own food, just like plants.
Wait, so cyanobacteria and cyanobacteria are different?
Yeah, I think they're different. So cyanobacteria is like a very simple kind of prokaryote. I think
So it's
cyanobacteria
even
are like,
simpler than bacteria.
I don't know if it's simpler. I think it's just considered to be different. Like cyanobacteria are one of the first things on Earth that perform photosynthesis. Right. So eukaryotes. I think we kind of pretty much talked about most of these things again, but just to repeat. So eukaryotic cells, right, are larger and more complex, right? So you have animal cells and plant cells, right? Now there are differences between animal cells and plant cells, but both of these are eukaryotic cells. Now in prokaryotic cells, generally prokaryotic cells they're usually unicellular. That means you have one cell, right? So usually like bacteria, like you don't have like a whole bunch of bacteria cells coming together and forming some kind of like super bacteria or something like that, right? Whereas in animals, right, like every animal is composed of like lots and lots of cells put together. So animal cells and plant cells, right, they can come together and form larger structures, tissues, organs, larger creatures, stuff like that. But bacteria and prokaryotes, they don't really do that. They stay unicellular. They stay by themselves. So even between plant cells and animal cells, there are some, differences, right? So these are both eukaryotes, but they have some differences between them. So animal cells have no cell wall, whereas plant cells do have a cell wall, right? Now, so plant cells have cell walls, animal cells no cell wall. Now remember, prokaryotes also have a cell wall, but usually the plant cell walls are usually more complex, right? Now plant cells also have chloroplasts, right, to make food, right? Well, obviously animals do not undergo photosynthesis, so they're not going to have chloroplasts, right? So chloroplast sounds kind of like chlorophyll, right? So that's where the chlorophyll is located, right? Now animal cells can have flagella, but plant cells, they do not have flagella unless, in most cases. They only have it in gametes. So in certain kinds of plants that live in the water and stuff like that, they could have a flagella. Now prokaryotes also have a flagella, right? But usually in animals, the flagella is usually more complex, right? So some of the things are similar between prokaryotes and eukaryotic cells, but even if they exist in both, usually in the eukaryotic cells, they're more complex, they're more developed. Mm-hmm.
Plant cell is a prokaryotic cell.
No. So both of these plant cells and animal cells, these are all eukaryotic cells. So here I'm just showing the differences between plant and animal cells. But all of these here are eukaryotic cells. So these are both different from prokaryotic cells. Prokaryotic cells are like bacteria.
So eukaryotic cells could be like unicellular and multicellular.
Correct, right. So eukaryotic cells, they could be unicellular or multicellular. So most are multicellular, only a few are unicellular. In terms of bacteria, or in terms of prokaryotics, they're all unicellular. So let's talk a little bit about cell membranes, right? So we talked a little bit about cell membranes before, right? So some of this is kind of a review. So in a cell membrane, remember, most of the cell membrane is made up of these things called phospholipids, right? So this was the structure that's kind of like fat, right? So do you see, each of these? You see each of these little circles with the two lines coming down, right? Right, like this tan circle with these two gray lines coming down, right? Each of those things is a phospholipid, right? Now, in the middle, let me ask you, like, why does the phospholipids arrange in this way? Like, why is it that all the circles are facing outside and all of the two lines are facing inside in the middle?
Whether because those lines are what's it called, hydrophilic, and those circles are hydrophobic. So you need the hydrophobic ones on the top so that things cannot enter inside.
Right. Close. So remember the inside is hydrophobic. Is that what you said?
Why is the inside hydrophobic?
So the outside, right? Outside of the cell membrane, is the outside environment mostly water or mostly lipids?
Depends, brother.
So it's
The outside
mostly water.
environment could be anything.
So it's mostly water,
Oh, all right.
right? Like the cytoplasm and the outside environment, most of the outside is water, right? Because right, so like most of the human body is made of water, right? So most of the outside is water. So the part that likes water faces the outside. The part that doesn't like water faces the inside and is protected. So you see all the circular little things, all these circular things all over the place. These are this is the hydrophilic part. This is the part that likes water. Inside the cell membrane, this whole portion inside is hydrophobic. It does not like water. Right? So the part that likes water faces the water outside. The part that doesn't like water is protected and faces only inside. Right? Now, inside the cell membrane, there's all kinds of other things. There's lots of proteins. You see these big chunks of things? Right? It says peripheral protein, protein channel, right? All these big, purple, blue, green kind of things, these are all proteins. So in the cell membrane, it's not just phospholipids, there's all kinds of proteins all over the place. Now, you also see inside of the cell membrane, you see these yellow things? See all these like little yellow things all in here?
Yeah.
All right, anyone know what those are? What are these like yellow things in here?
Lipids, brother. 100% lipids.
Lipids, right? Good. So those are lipids. What kind of lipids are these yellow things? Anyone recognize?
I think proteins are lipids or not? No.
So these are going to be.
I'm not sure about
So
that.
these are going to be cholesterol.
How do you know that?
So do you remember the structure of cholesterol? All those hexagons kind of put together. Right. So there's usually
Cholesterol is a carbohydrate.
no. So cholesterol is a lipid.
Cholesterol is a lipid.
So-
No, I don't remember, brother.
So cholesterol, right, or like steroid hormones, they all have kind of a similar structure. They all have like 3 or 4 like hexagons put together with like one pentagon at the end, something like that. So those are all lipids. So that means that they're going to be in the inside. They're going to want to stay in the inside in the hydrophobic kind of area, right? What about all this green stuff coming out? You see all these like green things coming out from the top? All these green projections,
Yeah.
right? Like all these green projections on the outside. What are these green things on the outside? Anyone know?
I don't know, brother. I don't know.
So those are all carbohydrates, right? If you look closely, all of those look like little hexagons. Remember how like monosaccharides, like glucose, right, fructose, all like little hexagons and pentagons, right? So these are all like a bunch of those like sugars put together, right? So the cells they have this the cell membrane is very complex, all kinds of proteins all over the place, cholesterol, sugars. All of this is used to communicate with the outside world and bring things inside when it needs to be, connect with other cells, all kinds of things going on here, right? Right. So most of this I think we kind of talked about, right? So it says here that proteins are important to cell membranes. So there's different kinds of proteins. So some of them are channels or carriers, right? So for example, this one over here on the left says protein channel. So you see there's kind of like a hole in the middle. So that's a channel for things to go in and out. So it can decide like what can go in and out, right? So for example, in this disease, cystic fibrosis, right? In cystic fibrosis, some cells have a defective protein, and it affects how salt moves across the cell membrane, right? So this protein, like it can, you can have problems, getting salt, outside of the cell, like putting salt and chlorine outside of the cell. So if this is damaged, then you have a disease, right? Now remember these green things, right? We talked about these carbohydrates, these green sugar chains outside, right? So that's what determines what blood group you have, right? So you have these different blood groups, right? A, B, ABO, right? So maybe you guys have heard of those, right? Like when someone donates blood or they lose a lot of blood, you have to get the right blood type, right? So the types of sugars that you have on the surface of your blood cells, that determines, okay, which of these blood types you have, all right? Now the cholesterol, right, it has an effect on fluidity of the molecules. So the more cholesterol you have, then it kind of helps to kind of break it up and make the cell membrane more fluid, right? So like lipids, right? Lipids can be like oily or they can be more solid, right? Like let's say you have butter, right? If you have butter and you put it in the fridge, it's like a solid, right? If you take it outside and put it, outside, it's going to melt and become like a liquid, right? So at the temperature range that human beings and the environment that we live in is at, right? Lipids are kind of right at the edge, back and forth, more solid, more liquid, more solid, more liquid. So you want to have a good balance, right? So cholesterol, right? That help, the more or less of that you have, it kind of changes it back and forth, more fluid, more solid, stuff like that, right? So the amount that you put there, and so different cells, they have different requirements. and so um, that changes the fluidity of the cells. So it's interesting. So they say that one of the theories, right? So like from salmon, right? So there's a good kind of fatty acid from salmon. Anyone know what that is? What is like the good kind of, or from like salmon, fish, stuff like that? Anyone know what that is called?
I don't know, brother.
Have you heard of like omega-3 fatty acids, something like that? Like
Brother, I've seen that in milk. I've literally seen that in milk. I've never
yes,
seen that in
yeah.
fish.
So some people they take like fish oil, right? So they can get that. So what they say, one of the theories as to why that's beneficial, is that when people have like heart disease, right? The arteries they get like stiff, right? And they're not fluid and flexible anymore.
Oh, so they
Right?
take more cholesterol that way.
So it doesn't have cholesterol, but what it is that this fatty acid, right, is very fish have developed this because they live in like cold waters and stuff like that, right? So in the
Mm.
cold, right, remember lipids they become more solid, so you don't want it to become
Oh.
solid, right? So fish have the kind of fatty acid
Mm.
in their cell membranes, so that it still stays fluid even when it's cold.
Mhm.
And so maybe that's why it helps in human beings, like when a person has more cholesterol, has closer to heart disease. This still helps to prevent that because it helps to keep it fluid, and it stops the stiffening of the arteries a little bit. So that's like one theory. So I think there's different experiments that show that maybe it doesn't have much of an effect in human beings. But it's like one theory that could is possible. So cells they want to keep everything in balance, right? The amount of sodium, the amount of salt inside the cell, the amount of water, the the temperature, it's all kept constant. So this keeping of things constant is called homeostasis, right? This word homeostasis. That just means that the different factors of a cell you want to keep it balanced. So this is giving an example at the level of the whole organism, right? So for example, let's suppose a person gets too cold. Well, what happens through the system? Um, it causes like shivering, for example, right? Or let's suppose a person becomes too warm, right? Well, then the blood flow near the surface increases, so that the body can help to kind of lose heat, or a person starts sweating in order to lose heat, right? So you can cool back down again, right? So the body cells.
Brother, sweat is just like cold. That's what helps you lose heat, right?
No, because so the water evaporates, right? So it's taking heat with it.
Oh, it takes heat from your body with it. I see.
Right, right. So at the level of the organism, at the level of the cell, you know, there's different things to just keep whatever it is, the pH of the cell, so that the inside of the cell doesn't become too much acidic or too basic. Um, so the concentration of salt inside the cell is constant. The concentration of potassium, all these different kinds of things that you want to keep about constant. So this process that the cell uses is called homeostasis, just to keep all these different kinds of things balanced and at a steady level. Right?
Brother, shivering works because your muscles like I don't know if they pump blood faster, but I know they move, which causes heat.
Right.
It's like when you work out or exercise and you're like your body gets warmer because your muscles are moving. So it's the
Right.
same thing with shivering, right? Your
Yes.
muscles
Yes.
move. So,
Yeah. So same thing, right? Just the movement is causing heat.
uh,
So cells are selectively permeable, right? So what does that mean? What does permeability mean, right? Anas, right? I think Anas is gone. Where's Anas? Right, so Anas,
Anas,
right, so Anas,
Yeah.
what does permeability mean? If I say the cell is permeable, what does that mean?
Like it can move.
Not move, not quite move. Yusuf, what do you think? What does it mean if I say the cell is permeable? What does that mean?
I'm not sure, brother.
Not sure. How about Freeha? What do you think? What does it mean if I say the cell is permeable to water? What does that mean?
Like resistant.
Resistant. So it means that the permeability is like how much things can go in and out, in and out. So cells are selectively permeable. That means that they don't just allow anything to come in and out of the cell. They decide, okay, what things should come in and what things should not come in, stuff like that, right? So there's different ways that the cells can bring things in and out of the cell. All right. So we're going to talk about this endocytosis and exocytosis a little bit later on another picture, right? But so there's a couple different ways in which cells do this, right? So first of all, this is just showing that when you have different kinds of molecules, right? Some of them can come in and some of them are not going to come in as easily. So it says over here that you have a membrane channel, right? So if you have a membrane channel that's specific to this one thing, well then that thing can come in easily through that channel. So if a cell, in order to get energy, cells have channels for glucose, for sugar. So when they need more energy, right, they open that up and more glucose comes in and they can use that to burn in order to produce energy for the cell, right? So this is a channel. Now, if you have a molecule that's more lipid soluble, right, like cholesterol, some kind of fat-like thing, okay, those kinds of molecules, they don't necessarily need channels. They can just come right through. You see this? It says lipid soluble molecules over here. And these kinds of molecules, they're lipid-like, right, that don't like water. They can just come right through. Right? And the reason is because you see this environment right here in the middle, right? This is a very hydrophobic kind of environment. And so that's just what these kind of molecules like, right? So they can just come right through.
Wait, brother, I have a question though. Since the middle part over here is hydrophobic, so the inside of the cell membrane doesn't have any water in it?
Right. There shouldn't really be any water here, right in the middle, right here.
Oh, okay. So there's only like stuff passing.
Yeah. There's like water outside here, and water down here, but not here in the middle.
Oh, I see.
So these anything that's lipid soluble, like for example, steroid hormones, right? Those hormones that we talked about, I think in the last section, when we talked about lipids, those hormones can just pass right through. Okay. However, let's say you have some hormones that are water soluble, right? They like to be around water. Those ones, they cannot just come right through. They're going to bounce right off. So they need specific channels to go through. You understand the difference, right? So if something is more lipid-like, it can just go right through the cell membrane. Okay. If something is more water-like water soluble, okay, likes to be around water, it needs like specific channels and places to go through, because it can't just go through anywhere. All right. Now cells, they can take things in and dump things outside, right? So this right here, this is called a cell membrane, right, on the outside, right? So let's suppose the cell wants to take this thing into the cell. So what it can do is it can form this small pouch, right? It can form like this small pouch and surround that thing and kind of take it inside. This is called endocytosis. Endo means like in, right? That prefix endo means like in. So endocytosis is taking this thing in. Let's suppose it wants to dump something to the outside, right? So if it wants to dump something to the outside, well, it kind of does like the reverse. Whatever this material is on the inside of this little circle here, right? It goes over here. It gets dumped outside, right? So that's called exocytosis, right? So endocytosis is bringing something in. Exocytosis is dumping something out. Right? Now, there's two different kinds of transport, right? So let's suppose you don't want to. Let's suppose apart from endocytosis and exocytosis, you just have like some small particles you want to bring in or take out. So there's two ways of doing it. One requires energy, and one does not require energy. Right? So you see over here, these two examples on the left over here. These are called passive transport. Passive basically just means that it just kind of happens, right? That's what passive means. So passive transport does not require energy. Right? Active transport requires energy. It requires, you see ATP, it requires this energy molecule to be used to force something to go through. So what do you think is the difference, right? So take a look at these examples over here. Can anyone tell me, like, what would make, like, why is that in some cases you don't need energy for the molecule to go through, but in some places it requires energy? Like, can anyone see like what's kind of the difference between the left two, and the right. Free, what do you
Brother,
think?
things like I would assume things like lipids and fats and stuff can easily pass through, but like if it was like water, soluble, the things that you're talking about before, like those would need active transport because you need a protein,
Hmm.
which would require energy for it to pass through.
Hmm.
Or like irons or stuff.
Hmm. Okay. All right. So, Anas, what do you think? What was your idea?
I said like if they're like irons or stuff which
Hmm.
require energy like to pass through, like
Hmm. Hmm.
there's also limited amount of space in the active transport.
Hmm. So, I think Free had it the most correct. So, you notice, right? So, all of these substances are coming inside, right? In this case, right? They're just showing it. They're all coming inside. Now, you see on the left over here, these two things. There's like a whole bunch of it outside, right? And
Hmm.
not much inside. So, if there's a whole bunch on one side and less on the other side, it's going to go from the place where there's a whole lot to the place that there's less. It's just like this. Like
Oh,
imagine just by because as they're
it's like
bouncing around,
electrons, brother.
yeah, it's kind of like this. Like, let's suppose you have like a whole bunch of people are showing up for like a gathering, right? They're gonna try to spread out, right? So if you have if like they're all gathered in one place, over time they're gonna want to kind of spread out and fill out the whole place. So they're gonna go from the place where there's like a whole lot of people to the place where there's like less people, right? But notice over here on the right, we're going in the opposite direction. You see, there's already a whole lot down here, right? There's already a whole lot of these green things. So if we want to bring in more green things, we have to like force it to come in. We have to use energy to force it, right? Let's suppose everybody's like kind of spread out, right? It's going to require some kind of effort to force them all together into one tiny space, right? That's kind of the idea here. So the idea is that things usually move from a place of where there's a higher concentration of them to the place where there's a lower concentration of them. So if you want to go in the opposite direction, well then you got to force it by putting in energy, right? So this one over here, if it requires energy, it's called active transport, right? Now on the left, there's two types of passive transport, right? This one on the left here, right, does not require a channel, right? That's just diffusion. That's called diffusion. If it requires a channel, right, like if it's a water soluble kind of thing, then this is called facilitated diffusion, right? Because facilitated means like you help something, right? Right?
Mhm.
Like if you say, "Oh, this person facilitated the process," right? That person is like helping in the process, right? These things are not just kind of diffusing through; they require this channel. This channel is like helping, facilitating the process. It doesn't require energy, but it just like creates like an open space where these things can go through, right? So these ones on the left do not require energy. This one on the right requires energy, right? Now within the passive though, right, there's two different types. One is like facilitated; it requires some kind of help, some kind of protein to go through. And on the left, it requires no help; it just goes right through. Right?
Wait, I didn't understand that one.
Hmm? Which one?
So for diffusion to lipid bilayer, like there's no energy needed?
Right. So both of these two on the left require no energy. That's why they're called passive. Passive means no energy required. Right? So those things, right? So I think we pretty much talked about those, right? So those things that are more water soluble, right? They're going to require like some kind of channel to go through, right? Now, when water goes through, so water has its own special kind of channel. It's called aquaporin, right? So this is the same thing, right? If you have a lot of water on one side, or I mean a higher concentration of water on one side, to go to the other side, water often goes through aquaporins in cells. So both plant cells, animal cells, they all have aquaporins. So that water, this is like a special channel for water, right? Now, let's just review like osmosis, right? What is the idea behind osmosis, Anas? What does osmosis mean? Like, what is this picture kind of showing?
This picture is showing how like how the cell reacts of water going in or out in like different
Hmm.
situations.
Right. So where it says hypertonic, right? What does hypertonic mean? Anyone know? Like Freeha, what does hypertonic mean? Like what is this environment over here?
It's like with water,
So
like
so
water is neutralized.
so hypertonic kind of means you could think of it like this way, like it's like very salty, right? Have you guys done those experiments where like you put like you surround uh you put something in like salt water, right? Like a flower or a plant or something like that, right? And then if you put in like in like salt water, like what happens to it? You know?
Wait, what did you say, brother?
So let's say you take a plant, right, and you dunk it in salt water. What's going to happen to the plant?
It's I think it takes some like I think the flower the plant starts like shriveling up
It's going to
like
shrivel, right? Right. So
yeah.
so so maybe you guys have done those experiments before, in like elementary school or middle school or something, right? Where like you right. So if you surround it with salt water, it's going to shrivel up, right? So that's what this is showing. Hypertonic means that the surrounding environment is like very salty, very high concentration of salt. Okay. The middle, isotonic, right, just means it's kind of like normal, right? Whatever's inside, the same outside. Right? So that way, so these blood cells, they're not shriveling and they're not expanding. Over here on the right, hypotonic. What does that mean, hypotonic?
The water does not have
Hmm?
salt at all or like not much salt,
What's
I
going?
would assume.
Yeah, you can use a Lysol thing. I don't. Oh, wipes. I don't think so. Sorry, what was it again?
I would assume it's like very less amount of salt or no salt at
Yeah.
all.
So this is like the opposite, right? This is like almost no salt, right? So whatever is the concentration of salt inside the red blood cells, outside is like it's like basically like just like water, right? So in that case, right, it's going to do the opposite. Instead of shriveling, it's going to expand because water is going to rush in, right? So this just like a review of osmosis, right? And so it's showing that so the water is going in and out through these aquaporin
Mhm.
channels.
Okay, so basically, brother, osmosis is the facilitated diffusion of water, and it's done through aquaporins, right?
Right, it's done through aquaporins.
So, is it only for water, or is anything that's like water soluble, like
Yeah, no,
something that wouldn't like
yeah.
you
So
know what I'm saying, like
I think aquaporins are only for water.
for water? All right.
Yeah.
And so then this reverse osmosis
Mm-hmm.
is I still don't get reverse osmosis.
So
No, there's no reverse osmosis. It's just that the water is moving different ways, right? So if
Mm-hmm.
it's a very salty outside, the water is
Mm-hmm.
going to move out. If it's like almost no salt, then water is going to move the other way.
So water moves where salt is.
Water move. Correct. Yeah, you can say that. So water
Yeah.
moves from less salty to more
Mm-hmm.
salty places.
Oh, so it's like in an attempt to balance.
Exactly.
That's why we need.
Yeah.
That's why we keep a lot of water in our bodies, right? That's one of the reasons to
Yeah, you
keep
can say that.
it hydrated, right?
Right. Yeah, you can
Basically,
say
like in this situation,
that. Right.
sure. Because then you would have a lot of salt content in your body, I guess.
Now, Freya, your question?
so osmosis like will need no energy to go through.
Correct. Right. Correct. So this one does not require energy, right? So it says facilitated diffusion, right? So remember that just means that there's a channel there that water can go through, but you don't need like some additional energy to be put in, right? So now active transport, I think we pretty much talked about this again. Active transport is like when you're putting in energy to move something across, right? So usually this is where you're trying to force something against its concentration gradient, right? In this case, right, there's already a whole lot of this substance outside. So if we try to like, whatever little is left inside, we try to push it out, then that's going to require energy, right? So it's giving you some examples, right? So this is one example, like a sodium potassium pump. So for example, sodium right, is mostly outside of cells, and potassium is mostly inside of cells, right? So naturally, like sodium is going to start to diffuse inside to try to balance it out. So you don't want that. So the cell uses this pump to kind of push it back out, push it back out, push it back out. So this requires energy, right? So this is just like an example. So a lot of ions like the sodium, potassium, these kinds of things, they always require energy to kind of maintain this separation. You don't want them to kind of just balance out and it's just the same amount inside and outside. You want like the outside to be a lot more or the inside to be a lot more. So to keep it that way, you got to keep energy into the system. All right. So I think that's